This conference will now be recorded.
Hello everyone, is my voice clear?
Yes.
Okay, thank you for the confirmation. A very good morning, good afternoon, and good evening for everyone. So let's start the today's sessions with the presentation from Dr. Lena Grace Beslin from Nesamony Memorial Christian College, India. Your slides are visible and you can start the presentation.
Can I see this? Okay, thank you. I'm Dr. Grace from Nesamony Memorial Christian College, India. Start off. My topic is marine natural products in drug discovery. Marine natural products in drug discovery. In our world, there is a continual need for new therapeutic agents, especially to treat a large variety of diseases. There are many number of diseases without effective therapies, such as cancers, viral and fungal infections, inflammatory diseases, and neurodegenerative diseases are nowadays not successfully treated. The development of resistance of pathogenic microorganisms to antibiotics and cancer cells to anti-tumor drugs requires the compensatory generation of new drugs. The notable microbes such as bacteria, fungi, aquatic creatures synthesize bioactive secondary metabolites known as natural products. The discovery of natural products from different organisms can be a great contribution to the field of drugs. The biological diversity of the ocean offers a vast area for the discovery of these secondary metabolites because representatives of every phylum are found in the sea. The ocean contains more than 2 lakhs described species of invertebrates and algae. However, this number is a small percentage of the total number of species that are yet to be discovered and described. Modern informatics and the increasing availability of the sequenced genomes offer more opportunities than ever for the discovery of novel bioactive compounds and biocatalysts from marine organisms. The identification of biosynthetic genes and gene clusters through the bioinformatics approach and their eventual heterogeneous expression will give a great impulse for natural product identification and drug discovery from marine organisms. Chemicals that are not needed by the organism for their basic or primary metabolic processes are called secondary metabolites. What are secondary metabolites? They are the chemicals that are not needed for the organism for its basic primary metabolic process. The natural products have been identified that inhibit cell division, the process that it is the primary target of many anti-cancer drugs. For the target of anti-cancer drugs, the product which are able to inhibit the cell division is taken as the primary target of anti-cancer drugs. The marine environment became a focus of natural products drug discovery research because relatively unexplored biodiversity when compared to the terrestrial environments. Marine sponges are among the most prolific sources of diverse chemical compounds with therapeutic potential. When we compare the therapeutic potential, the marine sponges are in a great place. 5,000 chemical compounds derived from marine organisms, more than 30% have been isolated from the sponges. There are two marine-derived pharmaceuticals that are clinically available today. What are they means? One is anti-cancer drug Ara-C. It is to treat acute myelocytic leukemia and non-Hodgkin's lymphoma. The next one is Ara-A. That is the antiviral drug used for the treatment of herpes infections. Both are derived from nucleosides isolated from shallow water marine spawn that belongs to the coast of Florida. Other marine sources of bioactive molecules with therapeutic potential are bryozoans, ascidians, mollusks, cnidarians, and algae. These are all having the bioactive potential. Then we can see about the successful drugs from marine environment. There are 7 therapeutic agents derived from the marine environment. Of these, 4 are anti-cancer, one is antiviral, one for pain control, and the other one for hypertriglyceridemia. Further, there are 13 agents that are in phase 1, phase 2, and phase 3 clinical trials. They reveal that mollusks, tunicates, and sponges as the richest sources of these valuable and bioactive substances. But it's shown that these organisms are often feeding upon the microorganisms that are the actual producers of the bioactive agents. Because these different types of sponges and other group of organisms feeding on the microorganisms. These microorganisms are the actual producers of the bioactive agents in their body. These microorganisms are regarded as a real metabolic jewel of the world ocean, especially the heterotrophic bacteria and cyanobacteria. About 80% of them are in clinical trials and approved pharmaceutical agents. We can see some of the important drugs, those which are derived from the sponges and other organisms. First one is cytosine arabinoside. This is the early marine drug discovery occurred from a Caribbean sponge. The name of the sponge is Cryptothecea crypta, which possesses metabolites with a very interesting but relatively simple modification of a nucleoside. The normal 2-deoxyribose ring of deoxythymine and uracil are replaced by beta-D arabinofuranase. In the place of 2-deoxyribose ring and deoxythymine and uracil, it is replaced by beta-D arabinofuranose. It was revealed that cytosine arabinoside was a potent disruptor of DNA replication. A particular beta-D arabinofuranose is a potent DNA replication that led to cellular toxicity, while the arabinoside derivative had antiviral effects. Then the metabolic backbone is acute myelocytic leukemia, later in antiviral chemotherapy. The next drug is echinacidin 743. This 743 is also known as trabectedin. It is a second discovery of unusual nucleoside sponge-based chemistry. Potent anti-cancer activity was detected in the extracts of a tunicate, Actinidia turbinata. It is sold under the brand name Yondelis, and this
Sorry
is
for the
used as an anti-tumor
interruption,
chemotherapy
Dr.
medication
Leena. Your
for the
PPT is
treatment
not
of
visible.
advanced soft tissues. You are not
You exited from share.
Visible. So sharing. Okay. Actually, ma'am, can I share the screen?
You have logged in two devices, so which one we should make the presenter?
Mm. Mm. You take the first one. Cut. നെറ്റ്വർക്ക് ശരിയല്ല. இது கட் பண்ணி விடு. ரெண்டு இது வருது. இதை கட் பண்ணி ரெண்டும் ஒன்று இதுல வருது. ரெண்டாவது கனெக்ட் பண்ணது முதல்ல ஒரு கட் ஆச்சு. கேமரா ஆனா பேசுறேன் இல்லை ஸ்கிரீன் ஷேர் பண்ணாலே போதும் நெட்வொர்க் சரி அவங்க கட் ஆகிட்டாங்க இனி அடுத்த ப்ரெசன்டேஷன் ம் 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 கட் ஆகி ப்ராப்ளம்ஸ் நெட் ஸ்லோ ஆகி போகிறது ஸ்க்ரீன் ஷேரிங் தான் போச்சு இடையில கட் ஆகுது இதுல 4 நெட்வொர்க் கனெக்ஷன் வருது
I think Professor Lena is having some network error. She will join shortly. Meanwhile, Yakin Liu from Woods Hole Oceanographic Institution, USA will present the talk. Hello, Miss Yakin, please start your presentation. We are waiting for that.
Oh yeah, sure. I'm supposed to start at 11:30, so I was wondering if I should wait 5 more minutes. Okay, I'm going to go ahead and start. Hi everybody. So my name is Yaqing. You can call me Marine. That's my nickname. I'm an assistant scientist from Wuzhou Oceanographic Institution. And today I'm presenting the work that I did with my colleagues Qingran Li from Clarkson University and with my colleague from Andrew Solo from Wuzhou as well. And in this study, we examined the effect of live coral loss on Great Barrier Reef fish stocks. So the motivation of our work is that climate change and coral bleaching has causing a lot of loss of live corals, and the predictions for the future of live corals is really doomed. So far, we have very limited empirical evidence on how was the effect of loss of live coral on the productivities of fisheries, especially for commercial fisheries, despite the fact that there were biological and ecological factors that has been detected. And this is an important question because there are 6 million reef fisher worldwide, and then 25% of them are subsistence fishers, and to which the coastal resources like coral reefs and the fish that depend on the coral reefs and as a habitat are very important for their livelihoods. We had a structure model that we started from a production function, and from there we generated the reduced form for the empirical analysis in this study. Pure theory in studying the habitat, environmental quality, and fish production has started from a long time ago. And there were pure theoretical work, and there are also models with simulations. Then what has been limited in the empirical study is really is an estimation of the impact, accounting for the dynamics of the fish stocks with as well as the dynamic changes in the environmental conditions. So that's what we're trying to achieve here and trying to contribute to, is to derive from a production function and get down to reduced form that can be estimated based on observational data. So we started with the classic Fox growth model, in which the change of the stock is related to the Fox growth function and the harvest function. And to introduce the habitat effect, we linked carrying capacity with coral reef coverage in the function. This is equation 3 that you see here. And then we substitute equation 3 back into the Fox growth model with the stock dynamics to obtain a function that links catch per unit of effort, which is U, with other biological and economical factors in this model, which includes fishing effort, catchability coefficients, internal growth rate, and of course carrying capacity. And carrying capacity is linked with live coral coverage. So from there, we derived a reduced form that we can sort out data catch per unit of effort on the left-hand side, and on the right-hand side of the reduced form equation, we end up with data on fishing efforts E and with live coral coverage A. And here the bar means each observation, each variable is annual average observation. And then we can use this reduced form to use ordinary linear squares estimation to estimate parameters indirectly, which indirectly because we are not directly solving each individual unknown parameters like growth rate or the gamma factor that links the carrying capacity with live coral cover. But as you see for the highlighted red formula of each unknown parameters, we can solve the unknown parameters after we estimate this equation with ordinary linear squares. So we used the data from collected by the Australia Great Barrier Reef, and we have a 30-year time series of live coral coverage that is divided into 3 habitats. And it's measured in percentage of coverage by the area surveyed. And we have annual time series of catch and effort that's from 1990 to 2022 as well. And we selected 9 most important commercial fisheries species. And as you see, the distribution of these fisheries in terms of the annual catch and annual effort is very dispersed. We includes species that has high catch, such as coral trout, but we also have species has a low catch, such as saddle-tail sniper. So we feed in our data into the reduced form, and we estimated and backed out the individual parameters. And the one parameter that we're most interested in is the habitat coefficient here, which is the gamma. Recall gamma is the coefficient links the coral cover with carrying capacity. So gamma measures how the changes on live coral coverage is affecting carrying capacity of each individual species here. And we found their gamma is significant at the p-value 0.05 and 0.09 for 2 species, coral trout and saddle sniper here, for all the 9 species that we have tested. And here the p-values is attained using a residual bootstrap. It's a standard procedure from Friedman 1981. And we have also compared the fitted values from alternative model captures and then to show. So this is our goodness of fate for the coral associated fish. In general, the model captures the trends well, and we also see for saddle-tail sniper, the spikes on the data on the change in the catch per unit of effort was not entirely or was not well captured in the model. And the reason is that there were management changes over time on saddle sniper, and this model did not incorporate changes on management. And we discussed that in the discussion section of the paper. So after we obtained the parameters of gamma, which links the live coral cover with carrying capacity, what does it mean to us? So we can use our reduced form function to simulate the maximum harvest level, which is presented by maximum sustainable yield and maximum economic yield here based on our estimated parameter gamma. So here's what it looks like. So for coral trout, we can simulate if the coral coverage changes, the live coral coverage changes from 5% to 30%. For those two species, coral trout and saddle-tailed snapper, how the maximum sustainable yield, how the maximum economic yield is changing over time. Here, we see the orange lines, which represents the maximum sustainable yield. For coral trout, is a concave function that is increasing with a decreasing rate as coral trout coverage increase. In other ways, which means when live coral coverage decreases due to coral bleach, coral trouts, the harvest is going to decrease in an increasing rate. And for saddle-tailed snapper, we have relatively a little bit more flatter line compared to coral trout. If you recall, that's because the parameter gamma as estimated for saddle snapper is slightly larger, just slightly larger than 1. That's why the line looks a little straight here. So if we look at the data, when we compare at a certain level changes of live coral coverage, what does that mean for the changes of maximum sustainable yield and maximum economic yield? What we see here is not an insignificant change. I mean, it's very significant changes on the catch. So for example, if the live coral cover decreased from 20% to 15%, then based on the Great Barrier Reef's estimation here, we're looking at 12% or 29% reduction at MSY and 29% or 69% reduction of marine maximum economic yield, MEY. This figure just shows the computation where we find how the equilibrium point of the marginal revenue, marginal cost, where the economic yield was identified. I'm going to state that. So in conclusion, generally, we find that the reef habitat effects, they have significant effect on certain commercially important fish species, but not all of them. So obviously there is a heterogeneous live coral covers impact on fish species productivity. And secondly, the both species that we found significant impacts, and they are both outer shelf. They're both outer shelf, primarily using outer shelf as their fish habitats. So it highlights the importance of reef conservation on the outer shelf of GBR. And we would like to highlight that we have detected observed heterogeneous effect in among different fish species when we consider the impact of coral reef on the fish productivity, and so that might give fisheries management a guidance of how when implement marine reserves management and which species we should pay more attention to when we consider live coral, like coral bleach's impact on the fish's productivities. And of course, there are limitations on our study. We want to highlight that we are trying to estimate the long-term effect, given that we have a 30 years time series data. But we still want to say 30 years may not be long enough to measure the real long-term effects, because the reef structures may still be there within the 30-year time period. And our estimation cannot say anything about if the reef structure disappears, what's the impact, because that may requires even longer observations. We did not consider temporary management changes, and that's what we saw in the fitness figure of the fish snapper. And we also did not model. We also did not model delayed biological effects in the term sense that our reduced form is a continuous time, and we could have considered delayed biological effects by adding delayed variables such as live coral cover from 3 years ago. But when we tried that mod, we have tried 2 years delay and 3 years delay. But in both delayed models, we did not find a significant effects on the delayed effects. So here we just omitted that section and did not include that in our study. So that ends my presentation, and this paper has been accepted for publication at the Marine Resource Economics. And if anyone have any questions or any thoughts and for a discussion, and really have to keep in touch. And my contact information is here on the last page. So I'm really looking forward to collaborating with everyone that who is interested in this topic or this type of research. Thank you very much.
I hope there are no questions. Thank you very much for your presentation. We are moving on to next presentation by Srividya from India. Hello, Ms. Srividya. Are you going to share the screen? Are you going to share the screen? But your voice is not audible. Please check your speaker settings. We cannot hear your voice. Can you please check your speaker settings? Hello everyone, please find that we have only 3 presentations left for day 2 webinar. One presentation is already done and we are left with only 3 presentations. So I request Ms. Srividya to start your presentation now. I request Dr. Adiyanka Arohian from University of Port Harcourt, Nigeria to present the talk now.
Hello, can you hear me? Hello,
Yes, we can hear your voice, but your screen is not visible.
I'm trying to show my
Please share
screen.
your PPT.
I'm trying to share my screen.
But it's not the page of PPT. It is some other page.
Eh, I'll go to screen, right? Screen now. I'm on screen. I'm on screen. Okay, can you see my screen now?
No, you are opening the go to page, which we can see that is gotomeetings.com page. Open your PPT, then click on share button. You can see three windows. Click on the window where your PPT is there.
Okay. All right. Can you see now? Hello?
Yes.
Hello? Can you see my screen now?
Hello.
Yes.
Yes,
Is it
please start.
visible now?
Yes.
All right. My name is Areyo Adeyanka Richard. I read my co-author Henry Okoa Adana. I will prepare this article titled The Impact of Climate Change on Aquaculture and Fishery Production in Nigeria. Physical, environmental, and then social, as well as economic stressors already have a challenge on Africa coastal zones, and this is being troubled and worrisome by climate change. And then based on this, there's a kind of long-run danger for fisheries and aquaculture in the sub-Saharan Africa. And in the next few years, if this climate change is not controlled, it will lead to a loss of aquatic uh, livestock or animals in the zone, that is in sub-Saharan Africa. And we see that over the next 50 to 100 years, the ocean and atmosphere will going to warm. Sea levels will rise as a result of glacier melting and thermal expansion. The pH of the ocean will drop because of more acidic and will drop into the ocean, which the ocean will not be able to absorb all. Not only that, Nigeria aquaculture and fisheries industries is facing a variety of effects, impacts from climate change that need for flexibility management techniques. In order to achieve sustainable development goals of no poverty, no hunger, outstanding health and well-being of people, combating climate change and livestock below the sea is essential to Nigerian economy and also as well as in food sustainability. This research utilized annual time series data from 2009 to 2022. This study investigates the effects of climate change in the productivity of Nigerian fisheries and aquaculture. The figure on the screen is climate change system, which show the interactions of the atmosphere, hydrosphere, geosphere, and biosphere, as well as cryosphere interactions. And then as seen on the screen, we see that there is an interaction within the land and the sea and the ocean, which is a perfect interaction. If there's any breach in between, it will affect the life in the ocean as well. Like biosphere, atmosphere interacts inside the sea. Ocean and atmosphere also interact. Change in ocean circulations also, and then atmosphere also interacts with everything. The main impact of climate change on fishery. 1. Change in aquatic ecosystem and the distribution of species. Rising in sea temperatures, change in precipitation pattern, and as well as rising in the frequency of extreme weather, like increase in flooding, will affect the aquatic ecosystem. Reduction in production in aquaculture is also another significant that affects seafood from aquaculture, which is susceptible to climate change, both in quality and then in quantity, which is a result of climate change. Acidification of the ocean. Aquatic life is seriously threatened by ocean acidification due to a lot of discharge from industries, from companies that affect the life below the sea, particularly those livestock that are vulnerable to their shellfish life. Social and economic ramifications. There are significant economic and societal ramifications when declining fish supplies, lower aquaculture production, and changing ecosystems come together. The theoretical framework behind this study is the theory of Milankovitch theory. And then we have three major principles. 1. Eccentricity, which is the form of Earth's orbit around the sun, which changes over a period of 100,000 years from almost circular to slightly ellipse, which can be seen on the figure on the screen, that is figure A of it. And then also ancient heat, which is obliquate. It's also a change that happened around 41,000 years. The angles of Earth's ancient tilt swing between 22.1 and 24.5 degrees. This affects the seasonality of the sea and then of the earth as well, which can be seen in figure as seen on the slide, which is figure B. Precession is the earth's rotational axis work, which shifts the axial tilt direction over a period of 26 years. The timing of the season is in relation to Earth's orbit around the sun. It's imparted by precession. So the theory behind this is this movement which affects the life below the sea as well. Climate change effects on fisheries and aquaculture productivity. 1. Temperature changes, acidification of the oceans, variation in sea levels at ocean. 1. Temperature changes. Fisheries and aquaculture are impacted by this warming in a number of ways. 1. In the distribution of species. When temperature increases, it affects some species of aquatic animals. It also affects the growth and reproduction rate. When temperature rises, it affects the growth and it affects the reproductivity ability of such species. Also, rise in temperature also increase disease and then parasites in the sea, which also have a great impact on the fish and then on the aquatic animals as well. Now, coming to ocean acidification, it's caused by rising carbon dioxide levels in the sea, and it has an impact on aquatic ecosystem. 1. On structures of calcium carbonate, and specific species that cannot produce their skeleton and outer shell due to acidification are mostly affected. For example, like corals and mollusks, they could not produce their skin when the acid level is high. Also, behavior of the fish. Fish migratory pattern and interaction between predators and prey is also affected due to acidification of the ocean. And then number 3, variations in sea level and ocean current. Sea levels have risen by approximately 10 to 20 centimeters worldwide during the 20th century, mostly as a result of thermal expansion, which include ocean currents, which affect aquatic life, migration patterns, and nutrient distributions of the lives that lie below the sea. Then sea level. Coastal fish farming equipment may be impacted by rising in sea level. The method we use in this study, that's methodology, as can be seen on the slide, Nigeria is a nation in West Africa within sub-Saharan Africa. The panel data utilized in this research span from the year 2009 to 2022. And then the data were gathered include aquaculture production, which is measured in metric tons, temperature, which is measured in degrees Celsius, then rainfall, which is measured in millimeters, and then carbon dioxide emission, which is measured in kilotons. Data were obtained from Statista, FO Stata, and then World Development Indicators, respectively. Model specification. The implicit form of the model is presented as seen on the slide, which is aqua production, temperature, rainfall, and then carbon dioxide. And then this model is also molded into a quadratic form, and then as well as into double log, as seen on the equation 3 on the slide. Then fully modified ordinary least square, FMOLS estimator was used following the SWAN 2022 and Oliver 2022 as well. And as seen on the slide, FMOLS is factor of estimated coefficient using the FMOLS estimator, while XT is matrix of independent variables for time period T. Then PT matrix is the matrix of weighting coefficient for time period T to address serial correlation. YT factor is the factor of dependent variables for time period T. YT is the number of time period in the panel. And then a fully modified ordinary least square was used to estimate the long-run impacts on the aquatic of the temperature, rainfall, CO2 on the aquatic production, while the Granger causality was used to measure the short-run relationship of the data. Critique, SPSS, and efficiency was used to analyze the data. Economic a priori expectation. Model estimation using the fully modified least square method. The coefficients are expected to look like this. That is, which is table 1, as seen on the slide. Temperature is expected to be negative. Rainfall could be positive, could be negative. And then CO2 is expected to be negative. Results. Seasonality of panel root test. Augmented Dick Fuller and Phillips Perron was used to test the stationarity. And then there's a mix of order 1, 0, which is level, and then integration of order 1 for all. Like aquaculture was looked at level and also at phase difference, both for ADF and PP. Also the same thing with temperature, rainfall, and CO2. This table on the slide is coloniality diagnosis of the data, which shows that there's no coloniality among the data since the variance inflating factors is just 1 plus, and then the tolerance factor is approximately equals to 1. If the variance factors is above 2, then there is a small coloniality. And then if the tolerance factor is not equals to 1, then there's also a presence of coloniality. But with this, there's no coloniality in the data, which helps us to further the analysis for the study. The summary of the data is seen on the slide. We saw the mean, the median, the maximum, the minimum, the standard deviation, skewness. And then looking at it, we see that using Jack-Barra probability, we saw that all the variables are within the mean of the data set used. Correlation test shows that CO2 have high correlation with aquatic production. This could be as a result of some of the aquatic that feed on the digested materials inside the sea, which is being helped by the CO2 itself. The panel fully modified least square, as seen on the slide, shows that rainfall and CO2 were significant. Rainfall is significant at 5%, but it's negative. This negative shows that if rainfall is reduced, it also affects the aquatic production. But 1% increase in CO2 increases the aquatic production, while increase in rainfall beyond the limit affects the aquatic production. Temperature in this result is a positive coefficient but not significant. The R square shows that 64.8% were analyzed in the data, and then the long run variance is 0.006, shows that there is a long run relationship between the aquatic animals and the variables used. Double watching so that the data is within the range, which is 1.95. The gram causality test, as seen on the slide, shows that there is relationship between the variable, that is between the temperature and aquatic production, and then like other, they were well related. That is, temperature also affects the aquatic production, and then temperatures also affect the rainfall, and then it also affects the carbon dioxide in the atmosphere and also below the sea. Analysis of variance shows that there is a relationship. It shows that temperature, CO2, rainfall have positive impact on aquatic production because they were well interrelated, as seen on the causality, Granger causality test here at large 2. Figure on the above shows the trend of analysis as obtained for the study. Then where it is marked with star red, show the current level of aquatic production in Nigeria, and then also with rainfall as marked, and then also with temperature, and then also with CO2. But looking at it, it was around 2015 that the aquatic was at the maximum. And looking at it at maximum here, the temperature was around 27.31, which is the average temperature that the aquatic animal need to survive. And also looking at the rainfall was around 110 ml, which is the actual rainfall that aquatic animals need to survive. Anything below this or above this will affect the aquatic animals. And then also with the CO2 is around 110, 110,000 kilometers, which is the average that the aquatic animals need to survive. Anyone above this affects the aquatic animal. That's why the graph started falling down to the present state of the production. The same thing as seen before. So this one shows the trend, now with mark of the moderate stability. To be sure that the data used is well stable, we run cumulative sum, and then cumulative sum of squares CUSOR, which is cumulative sum or recursive residual. It shows that the data is well used, which is between 5%. No data is outside the the boundary line, both for cumulative sum and then cumulative sum of square. Conclusion: fully modified ordinary least square shows that rainfall and carbon dioxide emission were statistically significant at 5% and at 1% respectively at long run, affecting aquatic production, because the ocean and the sea, they have ability to trap the temperature. That's why temperature seem not to be significant. However, temperature has a positive correlation on Nigerian aquaculture productivity, but is not significant. Gradual causality test shows the short-run causality for each and every variable, which shows the interaction between the carbon dioxide and temperature and carbon dioxide, and then rainfall and temperature. They are all well related at short run, affecting each other. The the limitation of this data is aquatic plant production were not considered, which also affect the production of aquatic animal as well. Necessary data is not available, and if data is available, it's better to analyze together to actually give the detail of aquatic production, because a sizable component of the aquatic aquaculture industry is also made up of aquatic plants. So better fishery and aquaculture productivity will result from the inclusion of this data for further study. Policy recommendation: stringent regulation should be implemented nationally to discourage intense aquaculture in regions with substantial carbon concentration and promote it in areas with deteriorated soil. Fish and aquaculture producers should take a holistic strategy that includes investing in technology which will reduce the impact of technology, implementing sustainable practices, and building climate resilience. Ecosystem-based adaptation should be incorporated into aquaculture strategy for management for priority resources use, ensuring that management measures are harmonized, and that one will reduce the climate change risk. In order to reduce the effects and boost resilience in both human society and natural ecosystem, adaptation to the current and projected climate change is very essential, which must be incorporated. Thanks for your audience. Hello? Hello? Hello?
Hello everyone. I hope my screen is visible to all.
No, we cannot see your screen.
Hello.
Hello, we cannot see your screen.
Is it visible? Is it visible? My screen is visible. Hello.
No, it's not visible.
Is it visible now? Is it visible now?
Yes, it's visible. You can start your presentation.
Thank you so much. Hello to everyone. It is a pleasure to be here with all of you today. My name is Srividya CP and I am from National Center for Aquatic Animal Health, Cochin University of Science and Technology, Kerala, India. And here I am excited to speak about my research on Daphnia embryonic cell culture, a revolutionary model for environmental and cellular science. Going to the introduction, Daphnia magna is an ideal organism for developing a cell culture system and standardizing transgenic experiments due to its ecological significance, genetic consistency, and lack of established cell line. The absence of established protocol for primary cell culture development and transgenic experiment in Daphnia species necessitate the formation of an optimized media and protocol. Additionally, the high sensitivity to environmental stresses emphasize the need for reliable in vitro system to investigate various biological phenomena, including its widespread application in toxicology. Here, we selected bisphenol A, hereafter BPA, to test the toxicity in developed cell culture system from Daphnia magna. Bisphenol A is extensively used in the synthesis of polycarbonate plastics, epoxy resins, and various polymer materials, leading to its widespread presence in the environment. BPA has been found to exert toxic effect on various aquatic organisms at relatively low concentrations. These findings emphasize the importance of detecting and mitigating bisphenol A pollution to preserve water quality and safeguard aquatic biodiversity. This research introducing a pioneer method by establishing a cell culture system derived from Daphnia magna embryos, offering a new approach to access transgenic studies and toxicity. These are my objectives of my study. First objective is to development of an embryonic primary cell culture system from Daphnia magna. Second one is the optimization of primary culture medium for the development of Daphnia magna embryonic cell line, and the optimization of transgenic experiment in developed primary cell culture. And the final objective is toxicity application in developed Daphnia cell culture. So the methodology I used of a primary cell culture system from Daphnia magna, I selected Daphnia having embryos at pre-blastula stages and surface sterilized, and egg were removed carefully without disturbing the gut of the Daphnia. After that, the removed egg is again surface sterilized with the same protocol used for the Daphnia surface sterilization and homogenized with a homogenizer. After homogenization, they centrifuged and resuspended in a media and seeded in a 96 well plate and incubated in 98 degrees Celsius incubator. Then optimization of primary culture medium for Daphnia magna. STT assay was used to perform used to analyze the impact of supplements in Daphnia embryonic cell culture system. That means whether this supplement is improving the cell culture or which has any negative or any positive effect on primary cell culture system developed. We checked different concentration of FBS, different type of carbohydrates, vitamins, amino acids, cholesterol, and selenium, and those effects were compared using STT assay. After that, subculturing method means both the trypsinization and strong pipetting were compared to finalize which one is better for Daphnia cell culture survival. And also BRDU assay was used to confirm cellular proliferation. The next objective was optimization of transgenic experiments in developed Daphnia primary cell culture. For this, we selected baculovirus system and we modified the baculovirus system and we got expression in the developed Daphnia cell culture. The methodology is, first we selected P-fast back transfer vector and modified this vector P-fast back transfer vector with a GFP from PEGFP N1 vector. So we cut the GFP from PEGFP N1 vector and inserted into P-fast back. So the transfer vector become P-fast back GFP. After that, into this P-fast back GFP, we modified this vector in two ways. One is with a BAM with a WSSV IE1 promoter, and second is with the IHHNV P2 promoter, which means that we modified this P-fast back GFP promoter, GFP vector with two different type of promoter. One is IE1 promoter from WSSV and next one is P2 promoter from IHHNV. After that, we got two different type of transfer vector, which is P-back IE1 GFP and P-back P2 GFP. Then this transfer vector we transfected into DH10 back E. coli, which contain backmid. With the help of this helper plasmid, this gene of interest is transposed into the backmid, and the backmid we modified with our IA1 GFP and with our P2 GFP. After that, this backmid were extracted from the recombinant backmid E. coli, and this backmid is transfected into SF9 cells. That means Spodoptera fungipora cell line, that is an insect cell line which is usually susceptible to baculovirus, to know whether the GFP is working or not. After that, after getting expression in SF9 cells, we again isolated the viral particle from this SF9 cells and then inoculated into the developed Daphnia primary cell culture to check whether any of these modified promoter containing backmid is working in our Daphnia cell line or not. And the last objective is toxicity application in the developed Daphnia cell line. For that, we selected BPA and we calculated 24 and 48 hour LC50 value of bisphenol A using STT assay, and with that concentration means 24 and 48 hour LC50 concentration, we did RT-PCR to analyze the gene expression. Going to the result section of the work, the figure A represents the Daphnia containing egg in the brood chamber. These eggs were we used for the development of primary cell culture system from Daphnia magna. For that, we selected the Daphnia with the embryos, surface sterilized and isolated, and as I explained previously, we did all the protocol and we developed a primary cell culture system from the Daphnia magna. Figure B represents the image of a embryonic primary cell culture developed from Daphnia magna. Then after that, we try different type of subculturing method. One is pipetting and one is strong pipetting and one is subculturing using trypsin. With the figure A and B represent 1 month old embryonic cell culture system derived from Daphnia magnas, and figure C and D represent the reattachment of cells after subculturing. Here, figure C represents the reattachment of cells after strong pipetting, whereas figure D represents the reattachment of cells after trypsinization. And the statistical analysis was done to know which method is better. So based on the percentage of cell attachment, from the figure also it is clear that the cell attachment is very less, but still the attachment is more if we are using strong pipetting instead of trypsinization. So we concluded that the subculturing method, strong pipetting, is better than trypsinization, but still very a few number of cells, that is less than 20% of cell is only reattached, and those reattached cell is eventually detached from the flask and dying after 1 to 2 weeks. So we can't subculture the cells, but we are able to maintain the cell line without subculturing for 2 months. So we try medium modification to increase the lifespan of primary cell culture. For that, we used 7 commercially available media and compared the effect of all this media with respect to the lifespan of the primary culture. We used XTT assay for all the comparison, and from the initial screening, we find out that Snyder's insect medium showing better results compared to others. Then next objective, we compared FBS, that is heat inactivated FBS and non-heat inactivated FBS, various concentration from 0 to 20. And third step, we tested different concentration of carbohydrate, and in the fourth step, we checked the different concentration of vitamin, and in the fifth, cholesterol, and sixth, different amino acids, and in the seventh, different concentration of selenium were we checked. From the comparison of FBS, figure A, it is clear that whether the heat inactivated and non-heat inactivated FBS has no significant difference between them, and it is showing that 5% FBS usage is 10% FBS usage is far better than 5, but has no further improvement if we are using 15 or 20% FBS. And for the carbohydrate, we got a positive sign from glucose, and the whole concentration that we tested showing there is no significant difference between the tested concentration of glucose. So the minimal concentration, that is 0.1, we selected for further study. Then in the case of vitamins, here we are getting a good division while we are added vitamin into the culture media. So we concluded that vitamin is needed for cell culture development from Daphnia magna, and different concentration from 0.1, 0.5 and 1% were tested, which is showing no significant difference between them. So as previously, we selected the lowest concentration -1 for the from this test. And next one is amino acid and cholesterol. There is no significant attachment or division. If we are adding amino acid or cholesterol into the media, there is no effect on cell division or attachment. So amino acid and cholesterol is not used or not selected for further screening. And the final screening was done with a different concentration of selenium, 0.01 ppb to 1 ppb. From that, 0.1 ppb is selected as it is showing higher attachment and division compared to the 0.01% and 0.01 ppb and 1 ppb. So the final medium consists of Snyder's insect medium with 10% FBS, 0.1% glucose, and vitamins, 0.1% selenium. Then next in order to check whether the cell is dividing or not, we did BRDU assay. We got positive signal from BRDU assay. Almost 40% of cell is showing positive result. So which means that this modified medium supporting cell division. Then for the transgenic study, as I told you as I explained in the previous slide, we modified baculovirus with two promoter. One is IE1 and another one is P2. We got a good expression in IE1 GFP, but there was no expression for P2 promoter. So we concluded that the WSSV IE1 promoter has the capacity to activate Daphnia cellular system and activate the translation in embryonic cell culture from Daphnia magna. So going to the toxicity application, we found that after XTT, 24-hour LC50 value of bisphenol A is 50 μm and 48 is 20 μm. We did RT-PCR to check mRNA expression of genes. We selected GPS, GST, MNSOD and CUZNSOD. We able to find that there is a fold change of expression in GPS, GST and MNSOD. All the selected gene is upregulated while we tested with both the LC50 concentration of bisphenol A. So the conclusion is that the work is concluded that we can able to maintain a cell culture system from the Daphnia magna, and we can able to perform toxicity studies using this cell line. Since in almost 10,000 works are published within a month in Daphnia magna, all work is limited to in vivo work. Why? Because there is no established cell culture system from Daphnia magna. That is the main motive behind to carry out this research work, and we were able to maintain the cell culture for 2 months, and we used this cell culture system as a platform to check the toxicity assay, to check the medium optimization, and also to check the transgenic studies. The study present a pioneer effort in optimizing cell culture medium and introduced baculovirus-mediated transduction studies in Daphnia magna embryonic cell culture. Additionally, these findings offer valuable insight for advancing crustacean cell line research and understanding biological process in controlled laboratory environments. We demonstrated that BPA can influence the transcription of gene associated with stress in response to Daphnia magna cells through an in vitro cellular assessment. Our finding contributed to a deeper comprehension of the cellular and molecular mechanism by which BPA operate in Daphnis. Furthermore, our result indicated the potential application of utilizing in vitro cell culture system of Daphnia species for various applications. Future investigation should focus on developing a Daphnia cell culture based toxicity screening platform for various chemicals, including bisphenol A or other toxicant exposure in Daphnis. These are the reference, and thank you. Section is open for discussion. If you have any doubt, please ask me directly. Any doubts?
I hope there are no questions. Thank you very much for your presentation. The next presentation is by Regina Neto.
Can you confirm if you are seeing my screen?
Hello everyone, my name is
Yes, we can see the slides.
talk to you about a very particular sponge that
And can you hear the voice in the video?
Yes.
Okay, thank you.
Hello everyone, my name is Regina Neto and I come from Portugal to talk to you about a very particular sponge that resembles a little mushroom called Artemisia transiens, about which there isn't much information about. So, to set the stage for how this project came about, I will start by talking where it all began, in the mesophotic zone. So, mesophotic ecosystems are marine environments found at depths between 30 to 200 meters. So, essentially, they exist between shallow ecosystems and the deep sea. So, at these depths, we find a very rich diversity of benthic life, with some species share with both shallower and deeper environments, while others are unique to this zone. So because of this, some researchers actually believe that mesophotic ecosystems might serve as biodiversity reservoirs because they are capable of replenishing shallower and deeper communities after disturbances. However, despite their potential importance to the health and productivity of marine ecosystems, these communities still remain relatively understudied. So there are significant gaps in our understanding of their unique species richness, their vulnerability to human impacts, and their current conservation status. Among the organisms that live here, sponges are one of the key components that contribute to the ecological balance of marine communities due to their abundance and species diversity. So these animals provide important ecological services like the biogeochemical cycling, water filtration, and even the creation of habitat heterogeneity. So their physical structure allows them to host invertebrates on their surface, for example, and provide habitats for commercially important fish and invertebrates. Additionally, these sponge aggregations can serve as nurseries and refuges for various species and act as substrate modifiers, as bioeroders and framework builders. Beyond this environmental importance, sponges and their associated microbial communities are sources of bioactive compounds with significant pharmaceutical potential, including antimicrobial, antiviral, and anti-cancer properties. So this makes them valuable resources with a high priority for conservation. So, unfortunately, like other important organisms in the mesophotic zone, including corals, sponges are highly vulnerable to the negative impacts of the fishing activity because it causes surface damage, dislodgement and sedimentation smothering, pollution and construction of subsea infrastructure. However, despite this acknowledged significance and the threats they face, our understanding of the factors influencing their abundance and distribution remains unclear. For that reason, the OSPAR Convention, which is a convention for the protection of marine environment of the Northeast Atlantic, the FAO, and the EU Habitat Directive propose to include these communities in the list of protected habitats and to be considered vulnerable marine ecosystems, with the ultimate goal to establish protected areas where these species thrive. Our mission was to study the mesophotic ecosystems of Viana do Castelo in the north of Portugal, and to achieve this, we used a remote-operated video, the one shown in this image, and we did 230 dives to explore the rocky substrates along the coastline within a 6-mile range and at depths between 3 and 110 meters. So through the video recording, we encountered large aggregations of the sponge Artemisina from different depths and habitats, and we were unaware of the existence of this species, and it had not been previously documented in northern Portugal. So the goals of this study were to present new findings of Artemisina in the Atlantic Ocean, provide a literature review to map the current distribution and ecological preferences for the Artemisina, assess the role of depth and slope in shaping the species distribution and abundance, identify spatial patterns in the species distribution, and also offer insights into the biodiversity associated with these sponge aggregations. So from the literature review, we found that these species only exist in the Iberian Peninsula, so Portugal and Spain. And the map shows that Spain currently has the sites with the largest aggregations here in red, particularly in the northern regions, and the Galician area. So in Portugal, we have significant aggregations found here in Viana do Castelo, here as a result from this study. So also according to the literature review, this species can be found between 35 to 127 meters. It prefers rocky substrates with some inclination, extending up to vertical walls, and is primarily associated with other sponges, corals, fish, and sea urchins and sea cucumbers. And also, it's very commonly found with these two species of sponge and coral. Regarding the habitat characterization, we aim to understand how slope and depth influence the distribution and abundance of Artemisina. To do this, we categorize the slope into 3 levels. So the first level is low, which includes flat areas up to 30-degree incline. The second level is medium, so slopes between 30 and 60 degrees. And the third level is high, which is between 60 degrees up to vertical walls. For the depth, we divide it into 5 categories between 30 and 80 meters. Finally, we look at associated fauna, which are the species that occur within or around the Artemisina aggregations. So for the variable slope, we can see here the 3 levels. So this will be an example of a low slope, a medium slope, and a high slope. And we discover some interesting findings. We observed that Artemisina was a preference for low slopes, which was surprising because most sponge species, including Artemisina in other studies, tend to favor inclined surfaces. So typically, sponges prefer these inclined surfaces due to sediment accumulation. So horizontal surfaces are more likely to collect sediments, which can smother the sponge and impede water filtration. So as a result, steeper slopes generally have greater abundance and diversity of species, leading to higher competition for space. So in our case, we can pinpoint exactly why Artemisina prefers low slopes, but there is a similar species called Suberitis carnosus that also favors low slopes. Researchers found that this particular species, that is also somehow mushroomy shaped, has structures like feet that keep it elevated from above the ground, so prevent sediment accumulation and allowing it to colonize substrates that other sponges might avoid. So this could be one of the reasons explaining these results. So regarding depth, we observe differences specifically in the shallower depth range. So although we lack insufficient environmental data to fully explain why Artemisina appears in significantly lower quantities at these superficial depths, we did notice from the videos that several interesting species of algae are still present at this level. So this suggests that competition could be a factor limiting their distribution in this range. In our study, we found Artemisina only between 30 to 80 meters, which represents the depth limit for this species in Viana do Castelo. However, we know that Artemisina can thrive at even greater depths. The reason we don't see them below 80 meters in Viana do Castelo is because of the bottom characteristic. So beyond 80 meters, the rocky substrates give way to a large stretch of sand, which is not suitable habitat for this species. So Artemisia needs hard surfaces to attach to, so the lack of rocky substrate is what limits the depth range here. As for the associated fauna, the results are congruent with the literature. Other sponges and cnidarians are the groups appearing most alongside Artemisina. However, we didn't find the association between Artemisina and this species of coral called Dendrophilia cornigera. And the reason we didn't find it's because of the geology of Viana do Castelo. So Artemisina only appears in the rock substrate near the coast, which is this gray area, and the coral Dendrophilia only appears in the rocky substrate that are far from the coast in this gray area. So this sand stretch, which is here in green, is limiting this species interaction. So lastly, we wanted to determine if the spatial distribution of this species actually followed a specific pattern, whether it was randomly distributed, clustered, or even dispersed across the area. So to investigate this, we conducted a Moore-Nye spatial autocorrelation analysis, which revealed a positive clustering pattern for this species. To actually pinpoint the areas where this clustering was most significant, we performed a LISA analysis, which is a local indicator of spatial association. So this analysis categorizes each site into one of 5 categories. For example, high means that a site with high abundances is surrounded by other sites with high abundances. High-low means that a site with high abundances is surrounded by sites with low abundances, and so on. In the context of our study, high sites represent hotspots for the species. So interestingly, the highest concentrations for Artemisina were found near the fishing restriction zone, where the underwater cable of the offshore wind farm is located. So it remains unclear if the absence of fishing in this area contributes to the species abundance, or if they simply thrive due to very specific favorable habitat characteristics. Therefore, the clustering observed in this specific area remains unexplained, mainly due to a lack of spatial and environmental data. So further research that integrates environmental factors is very crucial to better understand the drivers behind these clustering patterns. And this information is important because it helps identify priority areas for conservation. So, in conclusion, with this work, we extended the current range of Artemisina transiens. We found that in Viana do Castelo, these species prefer low slopes, which is not congruent with other studies and could be a reflection of the specific local environmental factors. It also appears more at depths below 40 meters and exhibits a non-random distribution. To wrap up, understanding this dynamic is very important for conservation efforts and the management of marine ecosystems, particularly in light of ongoing environmental changes. I want to thank you so much for your attention. I'll be happy to answer any questions you may have.
I hope there are no queries. Thank you very much for your presentation, Regina. And we are moving on to the next presentation by Suresh. And it was a recorded video presentation. I am going to share the video. Please go through it.
Okay, hello everyone.
ఎవరు మాట్లాడాలి?
And this is the Oceanography and Marine Biology Conference 2024. I am Suresh Falvia from Sri Lanka. This is an advanced review of the impacts of plastic pollution on marine environment and ecosystems. So we will see what about the content of this presentation. In general, the plastics are synthetic materials. They are synthesized using some organic polymers, and according to the modern world and demands, plastics have been become into replacement for some other apparatus such as the packing materials, electronic applications, water bottles, and covering materials. In generally, those plastics are lighter than metals and clay materials, and the handling is considerably convenient. That means it is bit easy, and also the usage of different types of plastics have been readily increased with those kind of merits. The accumulation and improper wastage have been become into a severe environmental issue because the plastics are naturally non-degradable material. The difficulties in the finding of proper discarding systems, especially in the domestic level. These are the major things regarding the problem generation when considering the discarding of plastics. The landfilling is the widely practicing method is most of regions, and direct disposal or improper disposal would be much harmful regarding each and every part of the earth. Also, the location of landfills nearby the oceans and following streams are dangerous because of the leaching of some toxic chemical compounds from such landfills are results of the incineration between water and waste plastics such as heavy metals and organic pollutants, and also the floating of plastic debris in different sizes such as nanoplastics, microplastics, mesoplastics, and macroplastics. Ocean would be some severe impacts on the marine ecosystems and living because of the tendency of the releasing of different toxic chemical compounds such as polyester into seawater during the slow degradation process. And also the floating plastic debris, that means lighter debris, create some habitations for various colonizing microorganisms, and ultimately the migrations of such colonies would be an effect of the equilibrium of such ecosystems. The absorption of some other pollutant presence in marine environment such as some persistent organic pollutants, that means POP, by the floating plastics and recognized as a threat for colonizing microorganisms. Also the proper dumping, reusing, recovering, recycling, and also the awareness of proper wasting of plastics are the common recommendations regarding the marine pollution consequence and the pollution control methods. Okay, these are the references, and also this is the end of this my presentation. If you have any questions, please contact me later using these email addresses. I will reply you. Thank you so much and have a nice event. Good luck.
Thank you very much, each and everyone, for joining the webinar on day 1 and day 2. So I'll be sending you the certificates at your email by end of the day, along with abstracts book. Kindly go through it. So this comes to the end of our day 2 webinar, and simultaneously we successfully completed going to complete the live conference in Barcelona, Spain in 3 to 4 hours. We have been fortunate to have a great identities from academics, industries, and other areas with us. We'll be glad and thankful if you could join us in our next conference in person or virtually. The dates will be informed to you by mail. Also, would like to request to provide your opinion or feedback with us. For any other queries, you can mail to the concerned program manager. Certificate will be emailed to you. Thank you and have a nice day.